Hello students, welcome to readmedprep Academy channel. Today in Principles and Processes of Biotechnology Part 3, we are going to talk about methods of gene transfer, screening for recombinants, genome sequencing and plant genome projects, evolutionary pattern assist using DNA, genome editing and CRISPR or Cas9 and RNA interference RNAi. After a recombinant DNA molecule has been generated, the next step is to introduce it into a suitable host cell. There are many methods to introduce recombinant vectors, and these are dependent on several factors such as the vector type and the host cell. For achieving the genetic transformation in plants, the basic prerequisite is the construction of a vector which carries the gene of interest flanked by the necessary controlling sequences, that is the promoter and terminator, and deliver the genes into the host plant. There are two kinds of gene transfer methods in plants. One is direct or vectorless gene transfer, and the other one is indirect or vector mediated gene transfer. First, the direct or the vectorless gene transfer. In the direct gene transfer methods, the foreign gene of interest is delivered into the host plant without the help of a vector. Common methods of direct gene transfer in plants are chemical mediated gene transfer, microinjections, electroporation methods of gene transfer, liposome mediated method of gene transfer, and biolistics. The chemical methods: certain chemicals like polyethylene glycol (PEG) and dextran sulfate induce the DNA uptake into the plant protoplasm. The second method is microinjection. The DNA is directly injected into the nucleus using fine tipped glass needle or micropipette to transform the plant cells. The protoplasms are immobilized on a solid support like the agarose on the microscopic slide, or held with a holding pipette under suction. Here you can see the cell is made stable and a microinjection containing the desired DNA is injected into the nucleus of the cell. The third method is the electroporation methods of gene transfer. A pulse of high voltage is applied to the protoplasm cells or tissues which make transient pores in the cell membrane through which the uptake of the foreign DNA occurs. Here you can see the method. Before pulse, the cell membrane is intact and the electrical potential is present. And when a gene of interest or the drug is introduced into the cell through the cell membrane, an electrical field is applied which alters the electrical voltage between the outside and the inside of the cell membrane and results in the entry of the genes into the cell. And after the pulse is over, the cell heals with the gene or the drug inside. Fourth method is the liposome mediated method of gene transfer. Liposomes, the artificial phospholipid vehicles, are useful in gene transfer. The gene or DNA is transferred from liposome into the vacuole of plant cells. It is carried out by encapsulated DNA into the vacuole. This technique is advantageous because the liposome protects the introduced DNA from being damaged by the acidic pH and protease enzymes present in the vacuole. Liposome and tonoplast of the vacuole fusion resulted in gene transfer. This process is called lipofection. Here you can see the liposome mediated method of gene transfer, where a liposome is taken and the gene of interest is incorporated into the liposome, resulting in the formation of a lipoplex, and this is introduced through the cell membrane peptidoglycan layer, which results in the formation of an endosome by a process called endocytosis. This endosome, when it enters the cell, it breaks down again and results in the release of the DNA and the liposome. And the endosome with the maturation occurs, resulting in the DNA fragmentation and results in the formation of a lysosome, which helps to digest the DNA and breaks it into fragments. What is biolistics? The foreign DNA is coated onto the surface of minute gold or tungsten particles of 1 to 3 micrometers and is bombarded into the target tissue or cells using a particle gun, also called a gene gun, or microprojectile gun or shotgun. Then the bombarded cells or tissues are cultured on selected media to regenerate the plants from the transformed cells. On the right side, you can see the gene gun method, where it has a barrel with a plastic disc with DNA coated gold particles, and the disc is topped by a screen, and the DNA coated gold particles are targeted on the plant cells and they enter into the plant cells. The other method is the indirect or vector mediated gene transfer. Gene transfer is mediated with the help of a plasmid vector. This is known as indirect or vector mediated transfer. Among the various vectors used for plant transformation, the TI plasmid from Agrobacterium tumefaciens has been used extensively. This bacterium has a large size plasmid known as the TI plasmid or the tumor inducing plasmid, and an aqueous portion is referred as tDNA or transfer DNA and is transferred to the plant genome in the infected cells and cause plant tumors or crown gall. Since this bacterium has the natural ability to transfer the transfer DNA region of its plasmid into the plant genome upon the infection of the cells at the wound site, it is also known a natural genetic engineer of the plants. Here you can see in the image, the Agrobacterium tumefaciens has a nucleoid within DNA, and the TA plasmid enters into the target plant cell on its own. Hence, it is called the natural genetic engineer. The foreign gene, example BT gene for insect resistance, and plant selection marker gene, usually an antibiotic gene like NPT2, which confers resistance to the antibiotic kanamycin, are cloned in the tDNA region of the TA plasmid in the place of unwanted DNA sequences. Here you can see the Agrobacterium mediated gene transfer in plants, where the Agrobacterium tumefaciens has the gene, and the DNA containing the gene for the desired trait is taken and is incorporated into the DNA, and this is done with using restriction enzymes and DNA ligase. And this plasmid with the incorporated DNA is introduced into the plant cells, and the plant cells will carry the tDNA carrying the new gene within the plant chromosome, and the plant cells are grown in cultures, and the regeneration of the plant occurs and the plant with the new trait is obtained. Now let us move on to the screening for recombinants. What is meant by screening? After the introduction of recombinant DNA into a suitable host, it is essential to identify those cells which have received the recombinant DNA molecule. This process is called screening. The vector of foreign DNA present in the recombinant cells expresses the characters, while the non-recombinants do not express the characters or traits. Some of the methods of selection are blue-white colony selection method, antibiotic resistant markers, replica plating technique, and molecular techniques. Now, let us discuss about the blue-white colony selection method. It is a powerful method used for screening of recombinant plasmid. In this method, a reporter gene LAG-Z is inserted in the vector. The LAG-Z encodes the enzyme beta-galactosidase and contains several recognition sites for the restriction enzyme. Beta-galactosidase breaks a synthetic substrate called X-gal. X-gal is 5-bromo-4-chloro-indole-beta-D-galactopyranoside into a suitable blue-colored product. If a foreign gene is inserted into a LAG-Z, this gene will be inactivated. Therefore, no blue color will develop. It appears white because beta-galactosidase is not synthesized due to the inactivation of LAG-Z. Therefore, the host cell containing the recombinant DNA form white colored colonies on the medium that contain X-gal, whereas the other cells containing non-recombinant DNA will develop the blue colored colonies. On the basis of the colony color, the recombinants can be selected. Here you can see in this image on the left side, the plasma vector is designed for the blue-white screening, where the plasmid will have an ampicillin resistant part, a lag promoter, multiple cloning sites, and a lax Z part and a ORI. When you take the blue-white colony selection method, the culture media will show blue-colored colonies with no inserts, white-colored colonies which contain the insert in an ampicillin IPTG X-gal plate. Now, what are antibiotic-resistant markers? An antibiotic resistance marker is a gene that provides a protein that provides cells with resistance to an antibiotic. Bacteria with transformed DNA can be identified by growing on a medium containing an antibiotic. Recombinants will grow on this media as they contain genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline or kanamycin, etc., while others may not be able to grow in this media, hence it is considered a useful selectable marker. Here you can see in this method the selection for plasmid uptake. So the antibiotic becomes a selecting agent. Only bacteria with the plasmid will grow on the antibiotic present plate. Here you can see on the left side where there is no ampicillin in the plate, all the bacteria will grow. In the plate on the right side, you can see the bacteria which have been transformed only will grow in an ampicillin plate. Now, what is this replica plating technique? A technique in which the pattern of colonies growing on a cellular plate is copied. A sterile filter paper is pressed against the culture plate and then lifted. Then the filter is pressed against a second sterile culture plate. This results in the new plate being infected with cell in the same relative positions as the colonies in the original plate. Usually, the medium used in the second plate will differ from that used in the first plate. It may include an antibiotic or exclude a growth factor. In this way, transformed cells can be selected. Here you can see the replica plating technique. A master plate is taken and a sterile velvet on a block is taken, and a replica is made on the fresh media, and it is inoculated into two plates, one that lacks ampicillin and one that contains ampicillin. Whichever plate lacks ampicillin, you can see the ampicillin sensitive colonies, and the colonies do not grow much on the plates containing ampicillin. Now, what is the molecular techniques like isolation of genetic material and gel electrophoresis? Electrophoresis is a method of separating the different biomolecules with positive and negative charges. The principle is by applying electricity, the molecules migrate according to the type of the charges they have. The electrical charges on different molecules are variable. So, positive charged cations will move towards negative cathode, whereas negative charged anions will move towards positive anode. The principles of two-dimensional electrophoresis is seen in this figure. You can see that the protein sample is taken with a variable pH of 9 to 4. At one end is 9, the other one is 4, and the separation in first dimension by charge and an isolate electric focusing is done. In the second stage, applying the first gel on top of the second cell is done, and at one end the pH is 9, the other end the pH is 4, and in the third stage they separate the second dimension by size. The size can vary from 220 kDa to 10 kDa by SDS electrophoresis. Now, what is this agarose gel electrophoresis? It is mainly used for purification of specific DNA fragments. Agarose is convenient for separating DNA fragments ranging in size from few hundreds to about 20,000 base pairs. Polyacrylamide is preferred for the purification of smaller DNA fragments. The gel is a complex network of polymeric molecules. DNA molecule is negatively charged molecule. Under an electric field, the DNA molecule migrates through the gel. The electrophoresis is frequently performed with marker DNA fragments of known size, which allow accurate size determination of an unknown DNA molecule by interpolation. The advantages of agarose gel electrophoresis are that the DNA bands can be easily detected at high sensitivity. The bands of DNA in the gel are stained with the dye ethidium bromide, and the DNA can be detected as visible fluorescence illuminated in the UV light that will give orange fluorescence, which can be photographed. Here you can see the working of the agarose gel electrophoresis. where the DNA sample is loaded onto the agar gel, and there is application of electric field, and the separation of the DNA molecules occur, and the DNA samples are collected, and UV transillumination is done and documentation is done of the DNA molecules. Here in this image you can see on the left side the bands of DNA in agar gel. On the right side you can see the gel electrophoresis instrument. What is agricultural diagnostics? Agricultural diagnostics refers to a variety of tests that are used for detection of pathogens in the plant tissue. There are two most efficient methods. One is called ELISA, the other one is called DNA probes. Now let us talk about ELISA. ELISA is expanded as enzyme-linked immunosorbent assay. ELISA is a diagnostic tool for identification of pathogen species by using antibodies and diagnostic agents. Use of ELISA in plant pathology, especially for weeding out virus-infected plants from large-scale planting, is well known. There are different types of ELISA methods. One is the direct ELISA, where the substrate and the primary antibody and the antigen is used. Indirect ELISA, where the substrate with the secondary antibody conjugate and the antigen is used. The sandwich ELISA, where the antibody is sandwiched between the substrate and the antigen. And the competitive ELISA, where an inhibitor antigen is used along with the substrate and the antigens and the antibodies. Now, DAS ELISA, double antibody sandwich ELISA. It was first described by Clark and Adams in 1977, where the antibody coated well is taken, it is washed well, and then the enzyme conjugated secondary antibody is added to the antibody coated well, and it is again washed, and the substrate is added, and the color is measured. Now, what are DNA probes? DNA probes, isotopic and non-isotopic, like the northern and the southern blotting, are popular tools for identification of viruses and other pathogens. Nucleic acid hybridization or blotting techniques. Blotting techniques are widely used as analytical tools for specific identification of the desired DNA or RNA fragments from larger number of molecules. Blotting refers to the process of immobilization of the sample nucleic acids or solid support like nitrocellulose or nylon membranes. The blotted nucleic acid are then used as target in the hybridization experiments for the specific detection. What are the types of blotting techniques? Southern blotting, northern blotting, and western blotting. Southern blotting, the transfer of DNA from agar gels to the nitrocellulose membrane. In northern blotting, the transfer of RNA to nitrocellulose membrane. In western blotting, electrophoretic transfer of proteins to nitrocellulose membrane. Let us talk about the southern blotting techniques. The transfer of denatured DNA from agar gel to nitrocellulose blotting or filter paper technique was introduced by Southern in 1975, and this technique is called southern blotting technique. The transfer of DNA to agar gel to nitrocellulose filter paper is achieved by capillary action. A buffer, sodium saline citrate, is used in which the DNA is highly soluble. It can be drawn up through the gel into the nitrocellulose membrane. By this process, the single-stranded DNA becomes trapped in the membrane matrix. This DNA is hybridized with the nucleic acid and can be detected by autoradiography. What is autoradiography? It is a technique that captures the image formed in a photographic emulsion due to emission of light or radioactivity from a labeled component placed together with unexposed film. Here you can see the diagrammatic representation of a typical blotting apparatus, where it has a plastic tray with the transfer buffer and a support block is placed on which a filter paper wick is placed. And a gel is applied and a membrane is applied and a rack of filter paper is applied and the paper tissues are applied. On the top is the glass plate on which the weight is placed. This is the apparatus as such. What are the steps involved in southern blotting technique? DNA is cleaved by restriction enzymes and transferred to an agar gel. The fragments are separated by gel electrophoresis. The gel is soaked in an alkali solution to denature the double-stranded DNA and then placed on the platform in a dish containing buffer. A membrane is positioned on top of the gel. Buffer drawn up into the top layer of the blotting paper passes through the gel carrying DNA onto the membrane. The DNA on the membrane is now fixed and is placed in a hybridization bottle with solution that contains a radioactively labeled probe and is gently rotated. The probe binds to the complementary DNA fragments on the membrane, and this is studied with autoradiography where the different size standards are mentioned, and autoradiography detects the fragments with the probe attached. Now, what is northern blot? It was found that RNA is not bound to cellulose nitrate. Therefore, Alvin et al. In 1979 devised a procedure in which the RNA bands are transferred from the agarose gel nitrocellulose filter paper. This transfer of RNA from gel to special filter paper is called northern blot hybridization. The filter paper used for northern blot is amino-benzyloxy-methyl paper, which can be prepared from Wattman 540 paper. Here you can see the technique where the sample containing the RNA is extracted and an electrophoresis is done and the RNA is separated based on the size, and the northern blotting transfer of RNA is done to the membrane, and the RNA is fixed to the membrane with the UV or heat, and the labeled probes are applied and the membrane is hybridized with the labeled probes, and the visualization of the labeled RNA is done on X-ray film. Now, what is this Western blot? Refers to the electrophoretic transfer of proteins to blotting papers. Nitrocellulose filter paper can be used for Western blot technique. A particular protein is identified by probing the blot with a radio-labeled antibody which binds on a specific protein to which the antibody was prepared. Here you can see the Western blot technique where the proteins are separated through SDS page. Proteins are separated on the gel blotted onto the nitrocellulose paper. The nitrocellulose paper is incubated with the patient's antisera or the first antibody, and the incubation is done with the enzyme conjugated antihuman serum or the second antibody, and the antibodies attached to the specific antigen bands are studied on the nitrocellulose paper, and the enzyme conversion of substrate identifies the antigen, and bands can be visualized of the proteins. Now, what are the differences between the blotting techniques? One column is the particulars, the southern blotting, the northern blotting, and the western blotting. The name, southern name of the inventor. Northern blotting, northern is a misnomer, western blotting, western a misnomer. So, southern blotting there is separation of DNA, whereas northern blotting there is separation of RNA. In western blotting there is separation of proteins. Denaturation is needed in the southern blotting and western blotting, whereas in northern blotting denaturation is not necessary. And the membrane used is nitrocellulose and nylon in southern blotting, amino-benzyloxy-methyl in northern blotting, and nitrocellulose in western blotting. In hybridization, the DNA-DNA hybridization is done in southern blotting, whereas in northern blotting, RNA-DNA hybridization is done, whereas in western blotting, protein-antibody hybridization is done. Visualization is done in southern blotting by autoradiogram, in northern blotting by autoradiogram, and in western blotting in the dark room. Now, the bioassay for the target gene effect. Target gene is the target DNA, foreign DNA, passenger DNA, exogenous DNA, gene of interest or insert DNA. All are the names of that target gene. That is to be either cloned or specifically mutated gene. Targeting experiments have been targeting the nuclei, and this leads to gene knockout. There are two types of targeting vectors. They are insertion vectors and replacement or transplacement vectors. Now, what are insertion vectors? They are entirely inserted into the targeted locus as the vectors are linearized within the homology region. Initially, these vectors are circular, but during insertion they become linear. It leads to duplication sequences adjacent to the selectable markers. Here you can see the equal expression vector. The expression vectors contain operand sequences that allow inserted DNA to be transcribed and translated. It also includes sequences that regulate turn on or turn off the desired gene. When we study, we have transcription initiation sequences, restriction site, ribosome binding site, transcription termination sequence, selectable genetic marker, example the antibiotic resistance, the ORI, and the gene encoding expressor that binds operator and the promoter, and the bacterial promoter sequences and the operator. So all these are present in the E. coli plasmid vector. Now the replacement vector, it has the homology region and it is collinear with target. This vector is linearized prior to transfection outside the homology region, and then consequently a crossing over occurs to replace the endogenous DNA with the incoming DNA. So what is this transfection? Introduction of foreign nucleic acids into the cells by non-viral methods. Here you can see the image where a portion of the DNA region is removed and is replaced by another complementary DNA into the original DNA molecule. Now what is this genome sequencing and plant genome projects? The whole component of the genes that determine all the characters of an organism is called genome, which may be a nuclear genome, a mitochondrial genome, or a plastid genome. Here you can see the plants have three types of genomes. The genome that is present within the nucleus, the genome that is present in the chloroplast, and the DNA that is present in the mitochondria. So what is the genome project? Genome of many plants contain both functional and non-expressive DNA proteins. Genome project refers to a project in which the whole genome of a plant is analyzed using sequence analysis and sequence homology with either plants. Such genome projects have so far been undertaken in Chlamydomonas, which is an algae, Arabidopsis thaliana, rice and maize plants. Genome content of an organism is expressed in terms of number of base pairs or in terms of the content of the DNA, which is expressed as C value. Now what is evolutionary patterns assessed using DNA? First let us see what is a cladogram. In recent years, the evolutionary relationship between the different plant taxa is assessed using the DNA content as well as the similarities and differences in the DNA sequence or the sequence homology. Based on such analysis, the taxa and the relationship are indicated in a cladogram. Here you can see the example of an animal cladogram based on the vertebrae, bony skeleton, 4 limbs, amniotic, egg, hair, eggs with shells. So the animals can be classified accordingly in the cladogram, like the sharks, the ray fish or the finned fish, the amphibians, the crocodiles, the birds, the rodents and rabbits and primates. Here you can see the example of plant cladogram where they are classified accordingly to the evolutionary sequences of the DNA, like the anthophyte, the netifer, the nepine and the cycads, having different groups like cycads, ginkgos, conifers, gnetophytes and angiosperm. The cladogram, what are the uses? They show the genetic distance between the two taxa. It also shows antiquity or modernity of any taxon with respect to one another. Now what is barcoding? Barcoding in books and items in the supermarket is done. This will reveal the identity of the book or the items as well as the details like price. Similarly, barcode in genetic term refer to the identity of a taxon based on its genetic makeup. In practice, it is an optimal machine readable representation of the data which describes about the characters of any plants or any objects. Here you can see the barcoding can be done of different animals and plants using the DNA. Based on the nitrogenous bases and their differences in the sequences. What is genome editing and CRISPR or Cas9? Genome editing or gene editing is a group of technologies that has the ability to change an organism's DNA. These technologies allow genetic material to be added, removed or altered at particular locations in the genome. A recent one is known as CRISPR or CRISPR or Cas9, which is short form of clustered, regularly interspaced, short palindromic repeats, and CRISPR is associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate and more efficient than other existing genome editing methods. Here you can see the gene editing that has been shown. A DNA editing technique called CRISPR or Cas9 works like a biological version of a word processing program's find and replace function. First, a cell is transfected with an enzyme complex containing the guide molecule and the DNA cutting enzyme. A specifically designed synthetic guide molecule finds the target DNA strand, and the DNA is cut by the enzyme off from the original strand, and the target DNA is again attached to it. You can see in the last part the amended DNA strand which repairs itself after the target DNA is fixed. What is the use of CRISPR? Rice was among the first plants to be used to demonstrate the feasibility of CRISPR mediated targeted mutagenesis and gene replacement. The gene editing tool CRISPR can be used to make hybrid rice plants that can clone their seed. Imtiaz Khan and Venkatesan Sundaresan and colleagues reported in a new study which clearly shows one can re-engineer rice to switch it from a sexual to an asexual mode. You can see in this image a new variety of rice created by CRISPR gene. RNA interference. What is this? RNAi. All characters of an organism are the result of the expression of different genes which are regions of nuclear DNA. This expression involves transcription and translation. Transcription refers to the copying of the genetic information from one strand of DNA called sense strand by RNA. Here you can see the DNA replication occurs resulting in the transcription of the RNA, and RNA replication occurs resulting in the formation of protein by translation. When the RNA forms a complementary DNA strand, this is called reverse transcription. The addition of RNA before translation. What is this? This RNA, as soon as it is formed, it cannot straight away be sent to the cytoplasm to undertake the process of translation. It has to be edited and made suitable for the translation, which brings about protein synthesis. One of the main items removed from the RNA strand are the introns. The RNA splicing in plants is done by the removal of the introns, and the method by which the introns are removed from the RNA strand is called splicing. What is RNAi pathway? All the changes of removal of the introns before translation normally takes place whereby certain regions of DNA are silenced. However, there is an RNAi pathway. The RNA interference is a biological process in which the RNA molecules exhibit gene expression or translation. This is done by neutralizing the targeted messenger RNA molecules. The first step of RNAi pathway, a simplified model of the RNAi pathway, is based on two steps, each involving ribonuclease enzyme. In the first step, the trigger RNA, either a double-stranded RNA or miRNA, primary transcript, is processed into a short interfering RNA, siRNA, by the RNase 2 enzyme called dicer and drosha. In the second step, siRNA are loaded into the effector complex RNA induced silencing complex, RISC. The siRNA is unwound during RISC assembly, and the single-stranded RNA hybridizes with messenger RNA target. This RNAi is seen in plant feeding nematodes. Here you can see in this image the dsRNA or the double-stranded RNA is attached with the dicer and results in an interaction between the dicer and the dsRNA, results in the formation of siRNA, which is taken up by the nematode. And the siRNA reaches the target messenger RNA, and RISC complex with the unzipped siRNA is formed, and this is cleaved again to form the small fragments of messenger RNA. This is done in the root knot nematode. So, we discussed today in the Principles and Processes of Biotechnology Part 3, methods of gene transfer, screening for recombinants, genome sequencing and plant genome projects, evolutionary pattern assessed using DNA, genome editing and CRISPR or Cas9, and RNA interference or RNAi. Thank you. Kindly subscribe, like, share and comment the channel readmedprep Academy. Log on to www.readmedprepacademy.com for MCQs. Our Facebook ID is readmedprep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is readmedprep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.